I've made dozens of videos and written about why the evidence for disease-causing particles termed viruses is lacking. However, when I cover one particular area in a video, there are always questions like, okay, I get that, but what about this? I think if you've seen all the relevant videos, read Virus Mania, and my other articles, then most of these questions are covered. But it seems that's too much work for some, so I've taken the challenge to produce a one-stop video. It appears that a 3-hour film can be summarised in a few seconds. Let's see if we can do a similar job with virology. What is a virus? The word virus derives from the Latin word virus, which simply means poison. So viruses are supposed to be bad guys. A virus is a tiny intracellular particle that infects and replicates inside a host cell to cause disease. Don't be fooled by anything less, otherwise we're not really talking about viruses. Like unicorns, many people talk about viruses, but no one has ever seen one in nature. There are sick people. There are times when people in one area get sick together. There are tiny particles everywhere. There are images of cells breaking down and vesicles. There are indirect techniques like the PCR and genomic sequencing. None of these things require the existence of viruses. Let's find out why. Viruses are part of the human imagination. What's making me sick then? This is a sick person. His doctor doesn't know what is wrong with him, so says it is a viral illness. The doctor was told by another doctor that people who are sick in this way have a viral illness. No viruses have ever been found in a person who is sick in this way, but appointment times are short and people need to be paid. This is people getting sick together simultaneously. Due to indoctrination and groupthink, many people believe that the only way this can happen is if a virus is passing between them. Many people got sick together in Chernobyl in 1986, and no, it wasn't a radioactive virus. There are many common factors that people in one place can share, such as the same water and food sources, the same air quality, similar sun exposure and vitamin D levels, shared psychological stressors. However, talking about such things could get you investigated by the authorities or your research funding cut. Some people seem to think that this is conclusive evidence that viruses do indeed exist.
Ooh, that's a bingo!
You fool! Don't you know they can grow viruses? When you've spent decades looking for viruses in people but can't find any, it's not a good look. Virologists needed to come up with something to show the audience. The electron microscope came into play in the 1930s, but direct visualization of any virus from a human was elusive. Then in 1954, renowned magicians John Enders and Peebles suggested that they had grown the measles virus in their lab. Let's have a look at their magic show by pulling back the curtains. First, they tried to produce alleged viruses from human lung cells, but nothing happened. Human intestinal cells, nothing. Human uterus cells, nothing. Human skin cells, nothing. Human muscle cells, nothing. Monkey nut cells, nothing. Human foreskin cells, nothing. Embryonic chicken cells, nothing. Things were not looking good, but then at last there was some action in the test tube with human kidney cells and rhesus monkey kidney cells. Now, measles is supposed to affect the respiratory tract in humans, but they had to jump organs, and then species to produce the now famous cytopathic effect. So how did the trick work? The patient samples were either blood or throat swabs immersed in cow's milk, then poisoned with penicillin and streptomycin. To the mix was added bovine amniotic fluid, beef embryo extract, horse serum, unspecified antibiotics, probably more penicillin and streptomycin based on Enders' previous chemistry experiments, and phenol red. The safari soup was then passaged, which means removing some of the cells and putting them into a fresh culture mix. This is another way to stress the cells, and the more it is done, the more it diverts from anything resembling what happens in nature. If the cells don't break down the first time, simply passage and poison until you see the desired results. Amazingly, this multi-stage toxic assault on the cells made some of them sick and look a bit odd under the microscope. To make these slides, they added formalin to the sample, then ethanol and ether, stained it with hematoxylin and eosin, and then washed it with 95% alcohol and oil, and topped it off with xylene.
That'll do, pig.
All perfectly natural, of course. And jackpot, said the virus hunters. This munted tissue is the work of a virus. It's a bit like detonating in a bomb and claiming that the subsequent crop failure appears to be due to pestilence. With a quick sleight of hand, the word isolation was inserted into Ender's paper, and because it wasn't being done according to the scientific method, there was no control experiment. Instead, Enders and Peebles broke the magician's code and nearly let the audience in on the trick with the following statement in the fine print. Cytopathic effects, which superficially resemble those resulting from infection by the measles agents, may possibly be induced by unknown factors. Subsequent virologists have concealed these details from the casual spectator and have been emulating the trick with more sophisticated sleights of hand. Using healthy cell lines that the invented virus is supposed to infect is for rookies. If you want lots of spectacular cell breakdown in test tubes, the experts now use reliable props, such as Vero cells. This is a monkey kidney cell line with abnormal numbers of chromosomes, found back in 1962. That's right, it's already defective before they help it become more defective and the cells start to pop off a few vesicles. Hey presto, take a snapshot of these babies and say that they are viruses. With no evidence at all, claim that the particles are infectious and kill the cell. Assemble some genetic sequences from the soup, then say that they are viral and came from inside the particle, with no step to prove this either. So that's how you culture a virus that doesn't exist. Add all sorts of biological soup to a cell line that is already abnormal and may come from the wrong species. Kidney cells are handy because antibiotics and other foreign matter are often toxic to them. If nothing happens, passage the mixture a few times to stress the cells to the max. Once the cells die as a result of the process itself, blame the virus boogeyman. Substitute the word isolation for something that means the opposite, so that biological supermix now means viral isolate. And ignore famous virologists when they suggest that they didn't properly isolate anything after all.
Well, what's the purpose of the purification then?
Well, to make sure you have a real virus, you know.
No need to ask, he's a smooth operator.
Messed up animal experiments. There's another part to the Enders measles virus invention trick, and that's when they tried to demonstrate that their biological soup was infectious and disease-causing. Now, the germaphobes claim that measles is one of the most infectious viruses, and it passes easily through the airborne route. So to provide a realistic model of this, they injected their biological brew right into the brains of some unsuspecting mice. Happily, on that occasion, the animals remained well, but in many instances, animals are tortured senselessly, and the anti-science sometimes goes off the gauge. In Virus Mania, we describe many of the bizarre experiments the virologists have been carrying out over the past century. For example, with regards to polio, we explain that Landsteiner and Popper chose to take a diseased piece of spinal marrow from a lame 9-year-old boy, chopped it up, dissolved it in water, and injected 1 or 2 whole cups of it into the abdominal cavities of 2 test monkeys. One died, and the other became permanently paralyzed. Their studies were plagued by a mind-boggling range of basic problems. First, the glop they poured into the animals was not even infectious, since the paralysis didn't appear in the monkeys and guinea pigs given the alleged virus soup to drink, or in those that had it injected into their extremities. Why did they resort to such Mickey Mouse experiments? Because contagion cannot be demonstrated in clinical experiments that involve alleged viruses, and this includes experiments with humans. The Spanish flu was supposedly caused by a highly contagious virus, except there is no evidence for this. In 1918, Dr. Milton Rosnow tried to infect 100 volunteers with the flu by spraying what he thought was the infectious agent into their nose, throats and eyes. This didn't do anything, so then volunteers spent time in influenza wards where they breathed in the fetid gas coming from very sick patients and were exposed to wet coughs right in their faces from multiple individuals. And not one got sick.
And no transmissions were made.
Every time these experiments have been attempted, they fail to show human-to-human transmission. I've followed the reference trails in my video, Secrets of Influenza, and it's a dead end. No confirmation of transmission can be found. Non-existent viruses, jumping species. Bird flu, swine flu, monkeypox, coronaviruses, and even HIV. Skip the part about proving that these particles even exist and are the cause of disease. When you don't even have evidence for the fundamentals, start to speculate about how they are apparently poised to attack us. In this crazy world, the virus hunters call for the slaughter of chickens, cows, or any animal suspected of harboring a virus.
What they have in reality in their test tubes, right, they have nothing.